Okay, good. So we have seen most of the pathways that we wanted to cover, right? We looked at your JAK-STAT, TGF beta, the SMAD ones, and then receptor tyrosine kinases, then hedgehog dependent, Wnt. Then last class we looked about the role of metalloproteases or ADAM10 and how it regulates the notch signaling, or for that matter the processing of some of those this membrane proteins like amyloid precursor protein and others. So together we saw these pathways. Now we'll just get back to this RAS MAP kinase pathway. I think most of you must have heard about it because it's very common pathway. Most of the receptor tyrosine kinases or most of the cytokine receptors, everything they will go and activate this MAP kinase pathway. So it's very common pathway that could get activated. Cool. So many years ago, so people realize that there is something like this small RAS protein. You guys know RAS is the GTPase, right? It's a GTPase, but it's not like G protein of your G alpha, G beta, G gamma. जो G alpha is there, no? GPCR signaling में जो G alpha है, that actually has the GTPase activity that can hydrolyze the GTP and what not. Like that, RAS also is a GTPase protein, but it binds to GTP, GDP. Its activity, whole activity is regulated by the, when it is bound to GTP, it will be of course active. When it is bind to GDP, it is inactive, right? So the bound nucleotide actually determines the state of activation for RAS kinases. So we will come to that part of it, but have you guys heard about certain mutations? So these RAS proteins, they are very famous. You know why? You must know by now why. Tanvi, do you know? Have you heard about RAS proteins anytime? Good.
Mostly this pathway is altered in cancer.
Very good. So there is HRAS, KRAS and NRAS, I guess. So they are mutated, right? So some of these cancer associated mutations, they render these RAS molecules, the protein, in a constitutively active state. What do we understand by that? Meaning, if GTP goes and binds to this RAS protein, there will be a protein called GAP. You guys know GAP? I think Aniket had shown once the GTPase cycle, no? So we have RAS. Huh? It was in the exam. Oh. So you have RAS, then you have RAS GTP. That's when your GEF will come. GEF is very good. Then this can, I am just drawing this RAS as a nucleotide free state. It is not bound to anything. But this RAS can become GDP bound when there is GAP, which is GTPase activating protein. In case of RAS proteins, this molecule becomes very important because these guys have extremely low, or for that matter no GTPase activity like your G proteins which are there in GPCR signaling. Those guys have very low intrinsic GTPase activity. They will hydrolyze GTP in some seconds, right? This guy will remain bound to GTP in the absence of GAP for at least 1 minute. So this 1 minute is nothing for us, right? But this 1 minute makes a lot of difference when we look at the spatiotemporal regulation in cellular context. When the things happen at nanosecond speed, 1 minute bound, imagine how many molecules it is gonna go and activate, Durgesh. You guys remember we talked about retina, the what is that called as rhodopsin and whatnot, and we remember there was this protein which was called as the gap for activation of the GTP, one of the intermediates, and if you have a knockout mice for that, 45-fold increase, 9 millisecond or something it happened, right? It was, and you can imagine the response will change. Same applies to this as well. So these proteins become very important. Now, as Tanvi said, these proteins are mutated in cancers. One of that mutation could render this RAS molecule in constitutively active form or constitutively GTP bound form, and that's when they will lead to the hyperproliferation and whatnot. So in this case, right, so if you have normal, you have RTK, gets ligand comes, binds to that, dimerization, phosphorylation, what not happens, it goes and activates your RAS molecule, right, which means that the activation of this RAS protein is dependent on your upstream signal. You guys are following what I said so far? Rishika, did you understand? Good. Now imagine there is a mutation in this RAS protein where it remains bound, to GTP, means it's not going to hydrolyze the GTP. In that case, even if you do not have this guy, still it will remain activated. Did you understand the difference? So the upstream regulation will be off. So these guys can act as a dominant negative or dominantly constitutively active, and then they can rescue the phenotype. Even if your RAS is not there, if you introduce this constitutively active form, without even activation of RTK, it can do the job. That's in general what happens. Many years ago, so most of our understanding of RAS MAP kinase pathway comes from the studies in Drosophila and C. elegans, nematode. Have you heard about C. elegans? Good. It's one of another very good model system. So in Drosophila, compound I. So if you look at this electron micrographic image on the left, that is the Drosophila compound I, and this is composed of 800 individual eyes called omatidia. So now we are trying to understand where does this pathway come from, how it was discovered, originated, or what we know today about this GTP binding protein called RAS, it came from. So this individual, these are like individual eyes omatidia. So each omatidium, it consists of 22 cells, 8 of which are photosensitive neurons called retinula, R cells. These are designated as R1 to R8. Do you see this cross section over here? You see R1, R2, R3, R4, R5, R6, R7, right? And there is this blue color cell kind of structure, that is R8. So that's how the structure, that's how they are arranged. Is it clear? And you guys can understand it is axons to brain because these are equivalent to the photoreceptors what we have, and photoreceptors are the neurons because they have outer segments and then the nerve terminus is connected to then there is synaptic terminus which is gonna go and optic nerve will come and then it will take it to the brain. Oh, what happened?
I don't know.
There are some insect. Oh, it's gone. उसको मार दो. दिशा मारो उसको. Oh, caterpillar. इसका कंपाउंड आई चेक करो. Good. And now we don't do those dissection experiments in college anymore, right? Frog and all that. You did? Zoology background you had? Physiology. Very good. Good. So this is how the structure of, so there are 22 cells which are called as R cells. Out of that there are 8 cells, R1 to R8, 1, 2, 3, 4, 5, 6, 7, 8. Okay. This is where we are. So, what people understood that there is a protein called this is a receptor tyrosine kinase family protein called 7-less. So, you will understand why these terminologies come from. Again, they come from Drosophila studies, right. The moment you mutate this protein, the development of these 7 cells will get affected. So, if that is why they named this guy as a 7-less or something of those sorts will happen. And it is very essential for development of the R7 cells, which essentially means that if you do not, that is when the name comes from. So, remember, this is going to be very important when we talk about the pathway further. 7-less is the receptor tyrosine kinase, okay, which is present on these cells. And in flies, flies with a mutant 7-less gene, the R7 cell in each ommatidium fails to form. Now, if you look at these images, this is your wild type image and you can see that 1, 2, 3, 4, 5, 6, this is the R7 cell, it is there. Whereas, when you have mutant, you do not see 7, right, everywhere you can see there are 6 there. So, you do not see 7, which means that this 7-less is very important to form. Now, the most interesting part about all this is that since this R7 photoreceptor is necessary for flies to see only in ultraviolet light, even if you mutate, these mutants can still see otherwise normal, they can normally they will see. Only when they are required to see in ultraviolet vision. I do not know when flies will go and see in ultraviolet vision, what is the context to that, but this is how scientists identified them. Therefore, fly R7 cells are ideal genetic system for studying signal transduction downstream of receptor tyrosine kinases. So, many of the times these systems were very helpful to understand the basic signaling pathways, not necessary that they have physiological relevance as such. I do not know when flies will see ultraviolet, but they have, this is the system what scientists were able to develop, that if you mutate this, because this is the receptor tyrosine kinase. Now, what you can understand, if you play with these receptor tyrosine kinase cells, you can understand what is the upstream of this molecule or downstream of this molecule and how it is going to be important in the developing of these R7 cells. Now, in this context, during the development of each ommatidium, there is a protein called Bb6, which is bride of 7-less, is expressed on the surface of this R8 cells. Now, you see, this is your R7 and this is your R8. R7 has receptor tyrosine kinase equivalent 7-less, which means this is the receptor. Who is the ligand for that? Is Bb6. That is why it is called as bride of 7-less. So, these guys are going to talk to each other. That is how it is. And so, R8 and R7 are placed like this, so that these two, the ligand and the receptor, can talk to each other. The extracellular domain of this membrane is the ligand for 7-less receptor tyrosine kinase. So, what people did is essentially they tried to understand how important these interactions are and what can they lead to the physiological relevance of this. They did some experiments where in wild type cells, what we know that if you have R8 cell which expresses Bb6, whereas you have R7 precursor cell which has the 7-less, these two guys come together. So, essentially there is going to be some pathway activation, right. It is the same receptor tyrosine kinase pathway. The receptor dimerizes, gets phosphorylated, and it will recruit something downstream that is going to relay the information to drive some gene expression somewhere. Is it clear? So, this interaction lead to the induction of maybe maturation of development of this R7 neuron cells. That is done. That is happening in wild type cells. When you have single mutant of 7-less, what do we mean that when you do not express the receptor tyrosine kinase? Are you following this, Teja? When you do not express receptor tyrosine kinase, even if you have a ligand, it is not going to be of any use. In that case, these cells will not mature. The photoreceptor equivalent cells will not mature. That is what the importance, which means that there is some role for this ligand receptor interaction. Is it clear? Now, what these guys did, before this experiment, they had performed some screen by using this system. They have a genetic system now, right, because you can specifically tinker with these R7 cells and what not. And Drosophila has been a great model to perform genetic screens where you can identify important proteins in any pathway. Like when people studied about secretory pathways, they use yeast, no, where you can, that's why most of the proteins are called as Sec61, Sec70 and all that, right. Similarly, in Drosophila, they did these genetic screens. When they performed these genetic screens, they identified there are 3 proteins which will be important in this pathway. One of that protein was RAS equivalent, another protein was GRB2 equivalent, and there was one more protein that I forgot. But remember, then they did this screen, they identified there is a RAS equivalent protein, SOS, that was SOS. SO is nothing but son of 7-less, that will come. Then they identified this, right. So, when they identified this equivalent, what they did? They expressed the constitutively active form of RAS in this 7-less background, which means that even if your 7-less is not there, still it will rescue the phenotype, which means the RAS is an important molecule. The activation of RAS is important for the development of these cells. Yes. These are proteins. I will come to GRBB2 now. I will come to them. All 3 will talk. We will talk about. I will come to that. Next slide is that only. So, essentially what we have understood that the RAS pathway was discovered in the during one example how it was discovered is the compound eye of Drosophila, where there are these R1, 2, 3, 4, 5 equivalent cells, retinal equivalent cells of photoreceptors, and the development of those cells will be dependent on the interaction between R7 and R8. R8 is a ligand, R7 is a receptor tyrosine kinase, and the downstream of this receptor tyrosine kinase is your RAS protein. This is clear, right? Now, how does this RAS get activated? That is when your GRB and other molecules will come. Now, you guys will go step by step. So, this is where they have given an example of FGF receptors, fibroblast growth factor receptors. So, as I said, RAS activation can take place by any receptor tyrosine kinases or cytokine receptors, and anything and everything which can upstream to that can lead to the activation of RAS MAP kinase pathway. So, as we know by convention, this is your inactive RAS. You see that this is this tiny black color anchor kind of thing you see, which is inserted in the membrane. It is a farnesyl anchor. So, RAS proteins undergo lipidation. So, there is a 15 carbon lipid moiety called farnesyl that is attached onto this RAS molecule that helps its insertion into the membrane. Remember that. When pathway is inactive, your RAS is membrane bound, but it is GDP. It is with GDP, right, Teja? Everyone? Pratibha, did you understand? Inactive RAS. Now you have fibroblast growth factor receptor next to it. What happens? Your ligand is coming, it goes, binds to this. Whenever ligand binds to the receptor, by convention, receptor tyrosine kinases undergo dimerization, and then they will phosphorylate the tyrosine residues, because these are receptor tyrosine kinases. These get phosphorylated. That is when your GRB2 comes. GRB2 protein is the scaffold protein. You guys remember when we talked about long ago JAK-STAT pathway, they had some SH2 domain containing proteins. SH2 domain everybody remembers by now, right, Rishika? They have the ability to go and recognize phosphorylated tyrosine residues, right. So, similarly this GRB molecule, GRB2, has 2 important domain, Durgesh, that we should make note of. One is your SH2 domain. Why SH2 domain? Because it can go and recognize the phosphorylated tyrosine residues. SH2 and SH3 domains. GRB2 is the scaffold protein, right? It has SH2 domain. So, what it is doing? It is going and binding to the activated receptor now. That is the first thing it did. Then it has SH3 domain which are in duplicate again. We will see the function of SH3 domain in 1 minute. Before that, so what is happening? The first thing which is happening is receptor binds to the ligand, gets activated, phosphorylated, then phosphorylated receptor is recognized by this protein called GRB2 through its SH2 domains. Sounds good? Then what happens? It recruits SOS which is son of 7 less, and this is the GEF. Now what the whole effort is going for what? To activate your RAS protein. How do we activate the RAS protein which is GDP bound? You exchange GDP to GTP. Now who is going to do that? SOS which is son of 7 less. How does SOS go and associate with your GRB2? Is through this domain called SH3 domain. Okay. SH3 domain, again they are very important for protein-protein interactions. Now this is the surface structure of this SH3 domain and how it goes and recognizes certain amino acid moieties present on its interacting partner. They can interact with multiple domains. In this case, for SOS protein, they go and recognize this proline rich sequence. You can see there are two prolines, proline 4, proline 7, and there is one arginine, right? And this is your SH3 domain. So the advantage of this is that this SH3 domains will have some kind of pockets or something where your proline heterocyclic ring can be embedded, and that is how it forms this. There is a kind of curvature to this slight tiny space where the peptide can automatically go and sit on this SH3 domain, and these heterocyclic rings can be embedded into these pockets, so that there is a stable association which will form between the SOS and your GRB2 protein. Is it clear? So GRB2 is a very critical molecule. In that case, if you remove GRB2, whole pathway cascade can be inhibited. Is it clear? You are following this, Sahili? So when you have receptor tyrosine kinase which gets phosphorylated, GRB2 goes there, recognizes the phosphotyrosine molecules through SH2 domain, then it binds to SOS through SH3 domain, and that is how it brings your GEF near your Vaishnav RAS, which is inactive GDP bound. Is it clear? Now what is happening? ECPC, very simple stuff. You have activated receptor, your GRB2, the GEF, and then GEF will lead to the exchange of GDP with GTP. Whenever GTP gets exchanged, your RAS will bound to GTP, gets activated and dissociates from the, of course, the whole complex, and it will get activated, and that is the next step is going to go and start activating something downstream. That is how it will give signal to the downstream molecules. Is it clear? Did we understand everything? Now let us start zooming in into how does RAS gets activated a bit more. So this is how your RAS molecule looks like. And if you guys remember long ago, again we saw a structure how these GTPases or GTP binding proteins are. They have switch regions, right? Switch 1, switch 2 and what not. In this case, there are two switch regions which are very critical. Switch 1 which is shown in this color, I do not know what color it is, blue color something. What is this color? You guys see better colors. Cyan color, okay. This cyan color is switch 1 and this magenta is switch 2 color. Sorry, cyan color, magenta is switch 2. Is it fine? Everybody can see this. Now, your GPP and PHP is nothing but when they resolve the structures, they use non-hydrolyzable forms of GTP. So that is that molecule, and this structure what you see is your GTP molecule, and there is a tiny magnesium ion. So remember, these magnesium ion are very important for stabilizing the GTP in the pocket with when it is bound to the small GTPases. Is it clear? Are we following? Everyone is on the same page? Good. So now there is some, this is how, and there are two molecules which are very important. In switch 1, threonine 35 and switch, yeah. So, stabilizing GTP. So, who? GTP goes and sits in between the switch 1 and switch 2 region, but magnesium ion stabilizes the GTP in the protein. And then you have switch 1, switch 2 regions, right. So, you have this glycine 60 and threonine 35, which are very important. There is a structure that I will come to next, this kind of spring kind of structure. You remember we had seen something like this, I will show you again that long back. So, this is how in general your RAS will look like that. If you start little bit zooming in, what we realize is that whenever you have this is GTP bound RAS, this is where switch 1, this is switch 2 and inside is your GTP sitting over there, which is only this red color tiny space we see. We will see little bit more of that here. Whereas in GDP, your switch 2 region is shown in green, whereas switch 1 region is shown in blue violet color and orange is your GDP. Do you guys see there is a difference between these two structures? Gaurav, what do we see? Position? Switch regions. Do you see the switch regions are slightly different in both the conditions, and this structure is overlap of both of them, which is trying to show. Now, if you look at this, so switch 2 region, which is magenta color here versus green color here. Now, you see your switch 2 when it is bound to GDP is slightly upward moved, whereas the magenta color is slightly different. So, they have completely different conformation as compared to when they are bound to GTP versus GDP, right. Similarly, in case of your switch 1 region also you see there is a change, and this is how actually it happens. So, your glycine 60 and threonine 35 switch 1 and switch 2 regions, they are kind of holding this is kind of spring, no, which is holding this GTP molecule like this, and the moment your what is that called as hydrolysis GDP will happen, the GTP, the third phosphate is gone, they will in relaxed position. So, it is like trying to hold it switch 1, switch 2, that is when these two switch regions are extremely important, and that is why this conformation might be there, whereas in other conformation they will look little bit away from each other. Is it clear? Now, this is very important when we are talking about, now we are saying how does RAS looks like when it is GTP bound, whereas it looks like when it is GDP bound. Who is going to do this exchange of GDP with GTP? Your SOS, that is son of 7 less. People have resolved the structure of this one as well. So, in this case, just look at the second structure, we should not get worried too much. So, this SOS has this alpha helix. Do you see this yellow color alpha helix? This alpha helix is sitting very nicely between switch 1 and switch 2 regions of your RAS. Okay. So, it goes and it sits into this switch 1 and switch 2 region in such a way that it is gonna destabilize the GDP association, so that GDP can be exchanged with GTP, and that is when once your GTP comes there, again the glycine and threonine, whatever two molecules, two amino acids were there, they will make a connection with that.
So, here SOS is playing the role of
GEF, yes, SOS is your GEF, that is how it is going to activate it. Is it clear? So, remember the conformation which is kind of GTP locked versus GDP locked. It's not very difficult, it's just that switch 1, switch 2 regions are very important to remember. Good, Gaurav, are you still with us? Perfect. Now, what we saw is so far the RAS introduction, how important RAS is. Now, let us get into the pathway little bit. Everybody can see this? It's little bit complicated. I mean, there are too many information, but we will go step by step. Again, same thing, you have your RAS, which is inactive GDP bound at the membrane. Then I am not going in details of how it gets activated. Essentially, there is some receptor tyrosine kinase or what not. They will come, get activated, GRB2 will come, recruit SOS, GDP GTP exchange will happen, and RAS will get activated. Now, what we see is the active RAS, which is GTP bound. Now, what is happening? So, there are three proteins, essentially three kinases which are downstream to RAS, and you people will have to remember this cascade always. So, whenever we talk about RAS pathway, you should be able to write all these details, but anyways there are three molecules. So, you have RAS which is GTP bound, it's gonna go and activate one kinase, this kinase goes and activate another kinase, this kinase goes and activate another kinase. This is the cascade which is gonna happen. So the first kinase which comes after RAS GTP is the kinase called RAF. Now let's see little bit about how does the RAF get activated. There are scaffold proteins called 1433 family proteins. These 1433 family proteins are kind of scaffold proteins. They are present in cytosol and they go and bind to multiple proteins and they essentially sequester them. Whenever any XYZ protein, any signaling protein which is not supposed to go and do any action, these 1433 protein go and bind to them. That's their job. Like that, they are going and binding to this inactive RAF molecule. How do they bind to inactive RAF molecule? Through its phosphorylation. So RAF, when your RAF is inactive, it is phosphorylated. And this phosphorylated molecule who is recognizing is your 1433 molecules. So essentially they are sequestering it in the cytosol. Now what happens when you have, and this inactive RAF, it has two domains. There is N-terminal regulatory domain and there is C-terminal kinase domain. It is a kinase. So essentially it should have a catalytic domain and it should have a regulatory domain, simple. So what happens, the moment your RAS gets activated, this RAF will move towards the membrane and it gets further phosphorylated, whereas what it happens is that its regulatory domain will go and bind to the active RAS. Because of that, the 1433 will dissociate from them and then there will be some activatory phosphorylations on this RAF molecule. In this case, if you pay a little bit close attention, right Anushka, because RAF so there will be another molecules that will take it to the membrane, that we are not talking about. So even that happens with AKT as well. There are so many proteins, they go and associate there. So what is happening is, maybe this phosphorylation which is there when 1433 is bound is inactivatory phosphorylation, whereas when it goes and associates with the RAS molecule, it will get activatory phosphorylation. Now they have not shown the receptor. It is possible that the receptor itself can phosphorylate this RAF molecule further at different sites. Is it clear, Durgesh? Did you follow this?
Does the RAF get activated after association with the active?
Yes.
This 1433 has usually two phosphate groups, but when it associates with active RAS, the second phosphate group is.
So that will dissociate. So there will be other activatory phosphorylations which are different. So if your RAS molecule, let's say, has serine, threonine, this is tyrosine. So serine, threonine, and then there are tyrosines which get phosphorylated, right? I do not know what exactly, I have not checked it. Hypothetically, it is possible that when it is bound to this 1433, the serine and threonine are phosphorylated, whereas when it goes and associates with the receptor, the tyrosine will get phosphorylated because those are receptor tyrosine kinases. That is how the differential phosphorylation might help in the activation of your RAF kinase. Is it clear, Durgesh? Good. Now what is happening? The moment your GTP is hydrolyzed, so that is the first job of your RAS. RAS gets activated and recruits your RAF kinase there. The moment RAF goes and associates with your RAS kinase, RAS GTPase, RAS का काम हो चुका है. It need not to do any other further job. What RAS is gonna do is, it will get GTP hydrolysis will happen. So whenever the GTP hydrolysis happens, the RAF will dissociate from RAS. So is it? Are you still with me, Teja? Is it too complicated? Sure? Okay. So we saw that there was upstream signal which came and activated RAS molecule. Whenever RAS gets activated, Vaishnav, RAS gets activated, it means it has bound to GTP now. Then somewhere in the cytosol, your RAF protein which was in inactive state, somebody was holding this protein in the cytosol so that it will not go and bind to the activated RAS, or it will not go to the membrane till it gets activated. These are this 1433. The moment your RAS gets activated, this RAF protein will go and bind to this activated RAS. The moment it goes, how does it bind to the activated RAS? Through its N-terminal regulatory domain, whereas its C-terminal kinase domain is still ready to do its job. The moment it goes and binds to RAS, RAS will hydrolyze the GTP. RAS will hydrolyze the GTP. The GTP hydrolysis will lead to the change in the conformation, which leads to the dissociation of RAF from the RAS. Right? Are you following? Everybody still good? The moment your RAF gets dissociated, which is active, your RAF is active now. This goes and phosphorylates a kinase called MEK kinase. Right? So, RAS activates RAF, RAF phosphorylates and activates MEK kinase. MEK kinase is an intermediate kinase which will further go and phosphorylate the final molecule called MAP kinase. What happened? अरे बोलो बोलो. This is the typical phosphorylation cascade which happens in the signaling pathway. This is a very typical example of that. One leads to the other, one leads to the other. Now, it can be taken in any sense. Okay, these additional intermediates, it can create a problem because let's say तुम्हारा MEK kinase mutate हो गया, it could create a problem. RAF kinase mutate हो गया, it could create a problem. So, it's possible that why can't your MAP kinase directly go and bind to that, right? Maybe you have, then this also provides an opportunity for the system to keep check on the activation and inactivation of this, right? Right? Because your RAS itself is regulated by GEF and GAP. Then your RAF protein comes. RAF are regulated by 1433, which means that there is additional kinase which is going to phosphorylate it, which will going to make it inactive. Then the moment it has to be activated, it has to be taken to the RAS. It will get activated when RAS binds to it. Then your MEK kinase comes. MEK gets phosphorylated. Then MEK is going to go and activate MAP kinase. And this MAP kinase, which is active, it can act. So it will have, it has, I don't know how many substrates it has, maybe 100, 200 substrates it might have. It can phosphorylate those many proteins, depend on the context. अभी एक्टिव है तो सबको जिसको मिलेगा उसको फॉस्फोरिलेट नहीं करना है, but it could do that. It can translocate even to nucleus also, and in nucleus also it can do some phosphorylations, or it can do some phosphorylations in cytoplasm as well. Is it clear, Durgesh? Did you understand? So remember this: RAS, RAF, MEK, and then MAP kinase. Yes.
There is another component called ER.
This is also same, the MAP kinase. Yeah. ERK. So people check ERK kinase. So this MEK kinase is nothing but the MAP and ERK kinase. So it goes and phosphorylates the downstream protein and activates them. So MAP is also called ERK only. So there is a family of kinases, right? MAP kinase, kinase, MAP kinase, 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 and then there are. You remember we talked about one such protein called TAK1. When we were talking about how your NEMO and UBK63 chains and IKK beta comes, what was the protein? That was TAK1. That TAK1 belongs to the family of MAP kinase, 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 I guess, right? Sorry? MKKK7 or something, it was MAP kinase 7 something it was called, no? I also forgot. मेरा भी शॉर्ट टर्म मेमोरी है, इतना याद नहीं रहेगा, बट राइट स्वामी विवेकानंद ने एक बार पढ़ा तो खत्म, रजिस्टर्ड हो गया. ऐसे होना चाहिए ना. You understand? So this MAP kinase will have so many substrates. So it has so many substrates. We'll see one example of that. So now you guys must have, this is by now everybody knows, right? When all of us we are doing 2D cell cultures, we always feed the cells with full nutrition, you give them serum, growth factors, whatnot, everything. You have to feed them well so that they will keep proliferating, right? The moment you remove your serum from the medium, what happens? Starvation. Then what happens? Have you guys done this experiment any time? I mean, Muskan has done it, but anybody else? You have done? What happens? Autophagy. Autophagy can be induced. Very good. What else? Round? Oh, how long do you do? That also, no? So autophagy also time dependent, right? So what happens to the cell cycle? Cell cycle. Does it get arrested in any phase? G0, G1 phase. It will not pass through G0, G1, right? Everybody understand? Have you guys done this kind of experiment? You guys have run PI staining anytime? This is your normal cell cycle profile, 2N, 4N. Have you seen this? And then the moment you add serum starvation, it might happen like this, where your sub G0 peak is going up. Maybe this peak will shift like this. You remember this? Because your cells need nutrients to grow. And the moment you add serum back to the cells, in flash of second, there will be whole transcription program that will get activated. So many growth factors will be there, no, in the serum. Everything will get activated. That's what the induction of quiescent cells with growth factor, it causes the expression of so many genes, and those genes are called as early response genes. And most of these early response genes are very important for cell proliferation, because cell wants to divide, make new cells and whatnot, right? One of the example of which these early genes is CFOS, which goes and activates some of other cell survival or cell proliferation or cell signaling proteins which are required for cell proliferation essentially. So, growth factor, if you add, the cells will start going. So, now what happens, how does this pathway get activated when you add this, right? Again, it is through MAP kinase only. So, we are not going to back to how gets MAP kinase activated. Essentially, some of these growth factors will have growth factor receptor. That growth factor receptor will be, Durgesh, protein tyrosine kinase, receptor tyrosine kinase, that will get activated, get phosphorylated. Then what it will do? It will recruit whom? GRB2. GRB2 क्या करेगा? SOS को बुलाएगा. SOS क्या करेगा? GF है, GDP to GTP, वो RAS एक्टिवेट करेगा. RAS एक्टिवेट होगा तो क्या होगा? RAF एक्टिवेट होगा. RAF होगा तो MEK kinase. MEK से MAP kinase. So now we have MAP kinase which is activated and it's a dimer. मतलब दो molecule एक साथ उधर है. It's not over. We have some more to talk about that I will cover in next class. So you have active and dimeric MAP kinase which is phosphorylated. So there is a mechanism of MAP kinase activation that I did not tell you, but essentially when the MAP kinase is inactive, its binding or binding pocket or active pocket is kind of shielded by some molecules, and the moment it gets phosphorylated, it just lifts up and then gets activated. That's a common mechanism for some of the kinases. I'll pull out that figure and then I'll show you next time how does it get activated. Now, as I said, this MAP kinase has so many substrates, right? And of course, this MAP kinase itself can travel to the nucleus as well. So, depending when and what is needed, it will do the job. So, as we are talking about induction of quiescent cells with growth factors, and it's going to give to the expression of some of the early response genes like CFOS and whatnot. So, this activated MAP kinase can do it by two ways. It goes to the nucleus and it will go and phosphorylate this factor called TCF factor, which is ternary complex factor. So, this is your CFOS gene which is inactive, where this TCF1 is inactive, means it phosphorylates TCF1 and this phosphorylated TCF1 will get activated. At the same time, in the cytoplasm, it will go and phosphorylate and activate this protein called P90RSK. I have no clue what does RSK stands for, but it looks like it is a 90 kilodalton protein which is there in cytoplasm and it is a substrate of your MAP kinase. Okay. Once this P90 RSK gets phosphorylated, it will enter the nucleus and it will go and phosphorylate this factor or co-transcriptional co-activator called SRF, the serum response factor, and then TCF and serum response factor both are phosphorylated, both are activated, will go and assemble at the SRE, means serum response element, which is required to drive the expression of CFOS and its downstream genes. Did you understand the क्या हुआ? Repeat करो. Oh, okay. So you understood how MAP kinase gets activated? How? बताओ, कोई जल्दी नहीं. हाँ. Perfect. So now you have active MAP kinase which is there in cytosol, right? ये सब तभी हो रहा है जब तुमने सेल्स को स्टार्व किया 24 घंटे के लिए, यू सर्व द सेल्स फॉर 24 आवर्स और अचानक से खाना दिया तो क्या होगा, वैसा हो रहा है. तो योर मैप काइनेज इज एक्टिवेटेड. मैप काइनेज does two things here to drive the expression of CFOS. CFOS is the early response element gene, right? So how do we do that? There are two things which are required to drive the expression of CFOS. One is your TCF, active TCF factor, and active SRF, which is serum response factor. These two has to be activated, and that's how your MAP kinase is activating the CFOS down expression. So MAP itself enters the nucleus and phosphorylates TCF, which is your ternary complex factor. At the same time, it indirectly activates SRF through phosphorylation of this P90 RSK kinase. It's another kinase. So this kinase, when it gets phosphorylated by MAP kinase, enters the nucleus. In the nucleus, it will go and phosphorylate this SRF molecule. So SRF and TCF both are phosphorylated, both are active, will assemble on your SRE. What is SRE? Serum response element. Right? And then once they assemble there, then they will drive the expression of your CFOS and all other downstream proteins. This is what I wanted to teach today. So there is two factors remaining. So there is a crosstalk between MAP kinase and GPCR signaling, which is required in yeast mating factors. There is one pathway that we will cover, and then I will touch upon the hippo signaling. That I will kept it for Friday, so that we could do our final last class on Friday, and then next Tuesday will be your presentations. Did you guys understand, by the way, do you understand this pathway? All of us, we are on same page with MAP kinase, right from your Drosophila, R1, R2, R3, R4, R5, R6, R7. R7 has 7 less, R8 has boss, which is bride of 7 less. Boss is the ligand. R7 is the receptor tyrosine kinase, right? The most important thing, if you guys think about it, even if you do not have upstream molecules which are activated, if you overexpress any of these downstream kinases which are constitutively active, they can still rescue the phenotypes. Did not understand. Did you understand? Like for example, see, so if you activate MAP kinase, or for example, if you take cancer, right? In cancer, there are so many mutations which happens. Some of those mutations can lead to the constitutive activation of these proteins, which means that even if you do not have growth factor receptor, even if you do not activate the growth factor receptor, even if you do not stimulate the cells with anything, if your RAS is active, it can lead to the signaling cascade, right? If suppose your RAS is also not active, but your MAP kinase gets constitutively activated. So, many of the times what I say by rescue means phenotype, meaning if you take quiescent cells, right, even without adding any serum to them, if you overexpress constitutively active MAP kinase, it should still lead to the it will still lead to the proliferation of the cells. Did you understand what I am saying, right? Like in case of your 7-less mutant Drosophila, even when 7-less was not there, if you overexpress constitutively active RAS, it still lead to the proper development of those cells, right? So, you have something upstream and then there is something downstream. So, if you activate this downstream pathway, you do not need upstream activation anymore. So, that is why these molecules become very dangerous in case of disease context, because any mutation, and this RAS is very famous, RAS, RAF. In case of melanomas, there is RAF mutations, which most of them they are known, right? In case of so many other cancers, these RAS mutations are known, and these mutations indeed can lead to the constitutive activation of these proteins. So, in any case, when you guys do these kind of experiments, I do not know how much you are going to do about, you are actually looking at the critical down signaling, hardcore signaling stuff, even if you do not do that, but you will always try to look for some of the rescue of one or the other phenotype, right? If you make a CRISPR knockout of your XYZ gene and then you reintroduce that protein, right? Do you do that? Complementation. No, you don't do. You guys are doing that kind of work? तुम नहीं कर रहे हो, करोगे ना कल जाके PhD में. PhD students, are you trying to do that? No. You guys are it's too early for you to think that. But hypothetically, Vaishnav, what happened? You should be doing so many mutations, no? तुम लोग करते हो ना RNA sequencing analysis. What do you learn from that? Dry lab. Oh, उसने बचा लिया खुद को. Easy peasy, yeah. But it is the way to do, right? That's the gold standard. You make a knockout, then you add your wild type protein. When you add wild type, you show that okay, this is specific. Rescued. There is no other molecule that is doing. There is not non-specific. Then what do we do? Sometimes these interactions, if you do mapping up two proteins, no, protein A, protein B's interaction is very important. Then you say that you disrupt the interaction and still you introduce, like for example, you have this RAS kind, you have this pathway, right? What is this called as? Let's say you take RAF kinase. This RAF kinase, I am giving you example, and this could become potential questions in your exams also, right? So you have RAS. This RAS is active. Then your RAF is coming and binding to this, right? Through this N-terminal regulatory domain, right? Now imagine you have mutation in this domain. What will happen? No, hydrolysis का सवाल ही नहीं है. So mutation can be two type, either gain of function or loss of function. It will either go and bind tightly that it will never come off, or it will not bind at all, right? So if you have to think about something like this, if at all you are asked or thinking about it, not asked, forget about it, in your project, right? When you are trying to understand there is a protein A, there is protein B, and then you show that these two protein interaction is very important to look at some phenotype, let's say cell proliferation, right? So you do hardcore biochemistry. I don't know nowadays people are not doing it so much, but people will map the domains. ये इसका कौन सा डोमेन किसके साथ बाइंड कर रहा है, उसका डोमेन इसके साथ क्या बाइंड कर रहा है. That thing is not done nowadays because we have a lot of funky techniques which people are using. But the typical way is you go and identify that one amino acid which is very important for the interaction. Right? Then you take that, yes.
How can you activate this downstream signaling by adding MAP kinase? It's a phosphorylation.
Arey, sir, 2 minutes, sir. It's almost. So it is phosphorylated, right? So MAP kinase will get phosphorylated. So your MAP kinase can get phosphorylated at 3 places: serine, threonine, or tyrosine.
You are externally adding.
You add it.
How can it enter inside the cell?
You transfect the cells, no? You clone it in the plasmid. The plasmid you put it in the cells, it will express. It's exogenous expression of your MAP kinase. And how to put it constitutively active? When you have serine or threonine or tyrosine residues, when a protein gets phosphorylated, what does it impart to that? अरे phosphorylation क्या करेगा प्रोटीन को? There are two possibilities. There is a phosphate group which is added onto the protein, and what does this phosphate group give to the protein? अरे एक्टिव ने क्या चार्ज देता है? Negative charge. So there are two things. So most of the times this interaction will be driven either through phosphate group or negative charge. 90% time it is not the phosphate group, it's the charge which is very important. So if you want to make any protein which is constitutively active, what do? You remove this serine or threonine and replace it with a negatively charged amino acid, so that it will mimic the phosphorylation, it will mimic that charge. Aspartic acid, there is glutamic acid also you make, and when you have tyrosine you make aspartic acid. So people based on the side chains and all, they do it. So if you take, even if you do not have anything upstream, if you take MAP kinase which is S2A or S2E mutant constitutively active and put it in the cells, it will still rescue everything which is downstream, ideally. If the phosphate is important, then it will not do. But mostly these interactions, these cascades go through the charge. Sounds good? Okay, so in next class we will try to finish up this and then we will touch upon the MAP kinase. Perfect.